In this lesson, we'll be talking about the Gram stain. So the Gram stain is a type of differential staining technique, which means you use two different dyes to stain different parts of cells or different types of cells. So it's much different than the simple stain where we only used one single dye to stain all the bacteria the same color and look at things like size, shape, and arrangement. And differential staining is more elaborate, and it's typically going to tell you more about the cell than just general physical characteristics. So with a Gram stain, it is a differential stain because it does require two different dyes, crystal violet as a primary stain and then safranin as a counter stain. And what it is going to do is it's going to tell us the different types of the differences in bacterial cell walls. So really what it does kind of do is exploit these kind of different types of cell walls. So with what we call gram-positive bacteria, when we Gram stain them, they're going to stain purple, and they have what we would call a thick cell wall. Cell walls are made up of peptidoglycan, so they have an abundance of peptidoglycan, which gives them their thick cell wall. Whereas gram-negative bacteria, when you Gram stain them, they stain pink. And they stain pink because they have a much thinner cell wall and have much less peptidoglycan. The Gram stain also can tell us about how many layers make up their cell envelope. So with gram-positive bacteria, which are here in the purple, they have two layers that make up their envelope. The inner cell membrane that surrounds their cytoplasm, and then their cell wall, which is much thicker and has the abundance of peptidoglycan. And then with gram-negative bacteria, which stain pink, they actually have three layers in their cell envelope. So they have the inner cell membrane that surrounds the cytoplasm. Then they have the cell wall, which has a thin layer of peptidoglycan. And then they get this bonus third layer called an outer cell membrane that sits outside of the cell wall. Now obviously when we're doing a Gram stain, we are not going to see these individual layers, not with our microscopes anyway. You would need like an electron microscope to see those. So since we can't physically see these layers, we do a Gram staining procedure and we can see the color differences and then just make assumptions based on the color that we see. So if we do a Gram stain and our sample stains purple, we know, okay, we're working with a gram-positive bacteria. It's going to have that thick cell wall and those two layers in its envelope. Whereas if the bacterial sample stains pink, we can just assume, okay, thin cell wall, less peptidoglycan, and has those three layers in the envelope because we have to account for that kind of bonus outer cell membrane. And this is just another image kind of looking at those layers. So again, gram-positive bacteria, when we Gram stain them, they are going to stain purple. Gram-negative bacteria, they are going to stain pink. And then here, if we look at the layers, so if this is a gram-positive bacterial cell, its envelope is only going to have two layers. So the innermost layer is the plasma or cell membrane, and that's just the phospholipid bilayer that surrounds the cytoplasm and is responsible for transport in and out of the cell. And then sitting outside of the cell membrane, that's where you have the cell wall with the very thick layer of peptidoglycan. That cell wall and its thickness is actually why gram-positive bacteria are able to stain purple, and we'll talk about that in just a second. Then over here we have a gram-negative bacterial cell. So again, innermost layer is the cell membrane. All cells have a cell membrane. It's selectively permeable and it controls what goes into and out of the cell. Outside of the cell membrane, we have a cell wall. So the cell wall is still there. It's just much thinner and has much less peptidoglycan. And then in gram-negative bacteria only, you get this third bonus layer called the outer cell membrane, which is still made up of a bilayer of phospholipids, just like the inner cell membrane. It's just called the outer cell membrane because it sits outside of the cell wall. So again, since we can't see these individual layers under the microscope, we have to do a Gram stain and then based on the color at the end we can make assumptions. So, in order to do a gram stain, first you would obviously need to go through the smear preparation. So you would need to add a drop of water to your slide, smear the bacterial sample into that drop of water. You would let it air dry and then heat fix it, maybe with a Bunsen burner or a hot plate, to stick the bacteria to the slide and to kill the bacteria and to kind of allow them to absorb stain. Then you would let your slide cool down, and then at that point you can go through and actually do the gram stain procedure. So this does have to be followed in this specific order. So it's always done in this specific sequence. So the very first step you would do is you would add crystal violet to the slide, which is a basic stain that's going to stain all the cells purple. You would add that to the slide and you would let it set for 1 minute. And at that point, all the cells, whether they're gram-positive or negative, they initially all stain purple because that's the only dye that you've used. So that sets for a minute, and then you just simply rinse with water. The second step is to add iodine, which we've done here. Iodine is not going to color the cells. Iodine just acts as like a sealant or a mordant and seals the crystal violet in. So it's just simply sealing the crystal violet in. We would let that set for 1 minute, and then we would rinse with water. At that point, everything's still purple. It's just sealed in now. Step 3 is the most important step. Step 3 is the alcohol or the decolorization step. So this is what we would call the differential step, because this is going to separate the gram-positive bacteria from the gram-negative bacteria. So how it works is you add alcohol to the slide, and this can be done several different ways depending on who's kind of guiding the lab and you're working with. So in this image it says to add this, the alcohol for 10 seconds. In my lab, I prefer to hold the slide at an angle and just add 1 drop of alcohol at a time and watch the dye run down the slide. And when the dye starts to run down the other end of the slide clear, then we stop adding alcohol and rinse with water. So there's several ways that you can do it, but regardless of the exact method, alcohol would need to be added to the slide. And then what's going to happen is, when alcohol is added to the slide, if your bacteria are gram-positive, because they have a thick cell wall, they will be able to hold on tightly to the crystal violet and stay purple. However, gram-negative bacteria had a very thin cell wall, so when they are rinsed with alcohol, it dissolves their outer cell membrane and then rips all the crystal violet out of their cell wall. They're not able to hold on to it because they have that very thin cell wall. They don't have a lot of peptidoglycan. So after we add the alcohol, and whatever technique we choose to do that is fine, but after we add the alcohol, the gram-positive bacteria is still purple, they held on to it, but the gram-negative bacteria are going to be colorless at this point because the alcohol pulled the crystal violet out of the cells. And the reason why this is called often the differential step is because this is at the first point where the gram-positives are going to look different from the gram-negatives, with the gram-positives being purple and the gram-negatives being clear at this point. So then after the alcohol, you would rinse with water. The very last step is to add the counter stain called safranin, which is just a pink basic dye that is going to stain anything that lost its color in the previous step pink. Because again, we can't see clear cells in the microscope, so we need to create that contrast. So we counter stain with safranin for 1 minute and then rinse it off with water. And at that point, your gram-negative cells are pink and your gram-positive cells are purple. Now there are some common mistakes that can happen when we do this because it is a series of steps. So if you mess up at any point, you can get kind of false results. Students will often say they don't see anything. That may be an error within the smear preparation. Maybe the slide wasn't heat fixed properly, so when the slide was subjected to the gram stain procedure and all the rinsing of the dyes and everything, it could have just rinsed off. So that's a common thing. What's more common though is missing the steps of the gram stain. So let's say you were going through the gram stain procedure and you completely forgot to do alcohol. So you did crystal violet, sealed it in with iodine, and then jumped straight to safranin. Well, if you forget to use alcohol, you do not pull the purple out of the gram negatives like you were supposed to, and in the end, all the cells will just look purple, which is obviously a false result if they were supposed to be pink and negative. Or let's say you forget to use iodine. So you do crystal violet and then go straight to alcohol and then do safranin. Remember, iodine is meant to seal the crystal violet in. So if you forget to seal it in when you rinse with alcohol, it'll pull the color out of all the cells. And then now, since the last step is to counterstain with safranin, all the cells will be pink, even if some of them were supposed to be gram-positive and purple. So you have to make sure you do the procedure exactly as is, or you will get these kind of false positive or false negative results. Another thing has to do with the age of the culture. So for the gram stain, you cannot work with a culture that's more than 24 hours old. We typically like to work with nice fresh cultures anyway, but it's particularly important for this one. What happens is if the culture is older than 24 hours, especially in gram-positive bacteria, what will happen is their thick cell wall will start to break down, and that will basically mean that they cannot hold on to the crystal violet like they should. So when they're rinsed with alcohol, they should stay purple, but if their cell wall has started to break down, they won't be able to hold on to the purple, and they'll actually stain pink in the end. So that can cause an error. A lot of times when we do that, we see something called a gram variable, where you see some cells that are pink and some that are purple, which typically means that some of the gram-positive bacteria, their cell wall was still intact, and they were able to continue on as normal with the gram stain and hold on to the crystal violet. Whereas others, gram-positive bacteria, if it was an old culture, their cell walls might have started to break down a little bit, which would inhibit them from holding on to the crystal violet, and then they unfortunately would stain pink in the end when they weren't supposed to. So it is important to work with fresh cultures that are no older than 24 hours.